What do you think of when you hear COVID-19? Do you think of the endless Zoom meetings, spending hours watching YouTube videos, dealing with your annoying siblings? Or do you think of the tiny culprit behind it all, the virus that causes the COVID-19 disease, SARS-CoV-2? Limiting the spread of this virus is one of the biggest challenges we face today. While washing your hands, wearing face masks, and social distancing are all important to do, hindering the reproduction of the virus inside someone's body is crucial to limiting its spread. So how exactly do we prevent the viruses from multiplying? Well, we should first understand how SARS-CoV-2 reproduces. After the SARS-CoV-2 retrovirus invades a host cell, its genetic material viral RNA is translated into viral mRNA, which can then be used to make the necessary parts of new viruses. Thus, the host cell is tricked into making more copies of SARS-CoV-2. But the cell doesn't just produce more viruses immediately. Ribosomes must first produce polyproteins following the instructions of the viral mRNA. Proteases, which are enzymes that help speed up proteolysis, the process of cutting proteins, must cut the ineffective polyproteins into functional pieces that form the viruses. But what if we could prevent the main protease, Mpro, from doing its job? An antiviral drug that serves as a protease inhibitor can bind with Mpro and prevent it from functioning. Then the uncut polyproteins would remain ineffective and in turn prevent viral replication. So how will the protease inhibitor bind to the main protease Mpro? You can think of the two components as puzzle pieces. In order for them to connect, their structures must be compatible with each other. Additionally, the protease inhibitor must contain a certain signaling molecule, or ligand, that alerts Mpro to bind with the inhibitor. After they bind together, Mpro will be unable to function, hence preventing the production of new viruses. This sounds a lot like trickery, but isn't that what viruses do in the first place? However, a danger of protease inhibition lies in the possibility of an antiviral drug binding to an incorrect protease, one that is necessary to the human body. Thankfully, the main protease Mpro SARS-CoV-2 has little in common with human genes and mutates slowly, so there is a very low probability of binding to an incorrect protease. Recently, scientists have identified 13 possible binding sites on Mpro and have measured the binding affinities, or strengths of the bonds, at each location with possible ligands. From their measurements, they distinguished the 4 best possible locations for protein-ligand binding, as well as possible protein-ligand crystal structures for protease inhibitor drugs to be modeled after. These findings provide a very useful foundation for antiviral drug discovery in the future.